Hello, this is Jesus Banales from Spain, and in this presentation I will introduce you the European Network for the Study of Cholangiocarcinoma, ENSCA, and our collaborative activities. ENSCA is an open, international, and multidisciplinary network of scientists focused on the study of cholangiocarcinoma. It was created in 2015 with the main objective to promote and boost collaborative research projects on cholangiocarcinoma at basic, translational, and clinical levels. This is a multidisciplinary network composed by basic and translational scientists, experts on cholangiocyte pathobiology, molecular pathology, stem cells, genetics, epigenetics, chemoresistance, and immunology, as well as clinicians, hepatologists, gastroenterologists, oncologists, surgeons, radiologists, and pathologists. Currently, 65 research groups belong to ENSCA, 57 from 15 European countries, and 8 groups from the United States acting as collaborators. Moreover, we are actively collaborating with different institutions, such as the European Association for the Study of the Liver, the EASL, the Spanish Association of Gastroenterology, the AMMF, the Cholangiocarcinoma Foundation, and the Global Cholangiocarcinoma Alliance. I show you here the members of the steering committee: Rocío Macías from Spain, Jesper Andersen from Denmark, Chiara Bracconi from UK, Domenico Alvaro, Vincenzo Cardinale, and Guido Carpino from Italy, and myself acting as coordinator. Our primary objectives are to follow up the natural history of cholangiocarcinoma in European countries, to improve the cholangiocarcinoma current classification by introducing new information on tumor morphology, pattern of growth, genetics, epigenetics, and molecular features, among others, to determine new risk factors involved in the development and progression of this cancer, to investigate on early and non-invasive tumor biomarkers for diagnosis and to estimate the prognosis, to study the molecular mechanisms of pathogenesis, to compare the effectiveness of the different therapies, and ultimately to select patients for clinical trials. ENSCA has a website, ensca.org or cholangiocarcinoma.eu. Moreover, we have also Twitter and Facebook profiles. In 2016, we published a consensus statement on cholangiocarcinoma in Nature Reviews. Last year, we published a special issue on cholangiocarcinoma with 11 review articles on different disciplines, and this year we published an update of the consensus statement in Nature Reviews. One of our first objectives was the creation of a European cholangiocarcinoma registry of patients, which is a fantastic platform for collaborative research projects. This project was competitively funded by the EASL in 2016 and 2019. We are using the RepCap online platform, in which we are including important clinical and biochemical data, general information on demographics, risk factors, and medical history, clinical parameters, biochemical and immunological, cholangiocarcinoma features are diagnosis, presentation, statement, treatment, and also the presence of all the kind of tumors. And moreover, we are including information of the availability of biological samples for collaborative projects. In parallel, we are doing an histological registry in which we are characterizing in detail the tumor tissue as well as the surrounding tumor tissue. The European Cholangiocarcinoma Registry is a retrospective study from 2010 and a prospective one, with the main objective to include around 4,000 cases by 2022. Currently, we altogether have included 3,592 cases from 30 European institutions. In general, most of the patients come from the Netherlands, Spain, Germany, UK, and Italy, but we also have cases included from France, Poland, Romania, Lithuania, Austria, and Norway. Several groups of ENSCA are also part of ESCALON, which is the European Latin American network for the assessment of biomarkers to predict and diagnose hepatobiliary malignancies and the characterization of risk factors for cancer development. This European Horizon 2020 project has 3 work packages on hepatocellular carcinoma, cholangiocarcinoma, and gallbladder cancer. In the work package number 2 on cholangiocarcinoma, we have the objective to create a Latin American registry of patients with cholangiocarcinoma. In less than 1 year, we were able to include around 100 cases from different countries in Latin America, from Ecuador, Peru, Chile, Argentina, and Brazil. In the registry, we have available information of samples in different laboratories that could be potentially used for collaborative studies. We have paraffin-embedded tissue, frozen tissue, DNA, RNA, urine, bile, plasma, and serum. In parallel, we are doing a cholangiocarcinoma histological registry coordinated by Guido Carpino. First, we are characterizing in detail 100 tumor tissue samples, paraffin-embedded, from patients that we have available frozen tissue, blood, and urine. The idea is to characterize in detail this tumor tissue at the histological level and then correlate the information with new information and data arising from the available samples at the genomic, epigenomic, or metabolomic level, for instance. The histological characterization of the tumor tissue is being done in collaboration with different pathologies of the registry. We are doing the cholangiocarcinoma histological characterization by evaluating the tumor cell differentiation, the proliferation index, the tumor stroma extension, the microvessel density, and the macrophage infiltration by using histomorphological routine stainings as well as different immunohistochemistries. All the images are digitalized, and every participant can access to them and evaluate them carefully. Some of the parameters evaluated can be also automatically analyzed by using different software and bioinformatic programs. ENSCA has 6 working groups on preclinical, histomorphological characterization, molecular profiling, development of novel therapeutic targets and tools, early diagnostic and prognostic biomarkers, and epidemiology, clinical characterization, and trials. Several groups of ENSCA are also participating in the first GWAS in cholangiocarcinoma, a project coordinated by Louis Roberts at the Mayo Clinic and supported by the Colangiocarcinoma Foundation. We all together were able to include more than 2,000 cases of cholangiocarcinoma and more than 500 cases of gallbladder cancers. The samples has been genotyped at the National Cancer Institute and the results are under analysis. Moreover, we are collecting new samples for a validation phase. Primary sclerosing cholangitis is a risk factor of cholangiocarcinoma. In this regard, there is an important international group, the International PSC Study Group, focused in the study of PSC. In this network, there are different working groups, and one is related to cancer. In this regard, we are deeply collaborating, ENSCA and the International PS Study Group, in order to see the similarities and differences of cholangiocarcinomas arising from a PSC background or for other etiologies. In 2019, we applied to a European Horizon 2020 cost action that has the aim to create European networks on science and technology. We applied with 13 European countries, 41 institutions, and 87 proposers, and had the support of the Colangiocarcinoma Foundation and the AMMF. Fortunately, this project was awarded, and we have a 4-year grant to carry out different activities related to cholangiocarcinoma. The chair is Vincenzo Cardinale, and I am acting as vice chair. We have different activities: management committee and core group meetings, working group meetings, workshops and conferences, dissemination programs, training schools, and short-term scientific missions. We had the first management committee in Brussels last March in 2019, where we define the different activities to be done in the next year and also in the 4-year grant. I show you here the grant diagram with the different activities proposed. In more detail, we first created a website which is called eurocolangionet.eu. Here you can find all the information related to the action. I show you here the action leadership position, so the leaders of the different working groups of the action, as well as the supporting committee. We had the first working group meeting in Malta last September, where we discussed the different collaborative activities to be developed and where we have the presentation of the different proposals. One of the first collaborative activities was the creation of a training school on experimental models of cholangiocarcinoma. This training school was going to be chaired by Chiara Bracconi and Diego Calvisi in Glasgow, but unfortunately, due to the COVID-19 pandemic, it has been rescheduled for next year, 2021. We also have a mentorship program, a program with the main objective to promote and help the career of young fellows. Moreover, we also have short-term scientific missions, so we have provided travel grants to different young fellows in order to visit different laboratories and to learn techniques and establish new collaborations between laboratories. We also created a COVID-19 task force group with the main objective to see the impact of COVID-19 in the management of patients with cholangiocarcinoma. And also in the website, you can find different job and training opportunities. We also generated a bi-monthly newsletter where you can find an update of all the activities of the COS action. The ENSCA has a biannual meeting. The first one was celebrated in San Sebastian in 2016. We had attendees from all over the world and a very nice scientific and social program. The second one was celebrated in Rome in 2018 and was organized by the group of Domenico Alvaro. As you can see, the number of attendees was significantly higher. And the third one will be celebrated in June 2021 in Edinburgh and is organized by our UK friends, mainly Luke Bolter, Tim Kendall, Juan Vale, Angela Lamarca, John Briswater, Chiara Bracconi, and Sahid Khan, and has the strong collaboration of the AMMF. ENSCA is also an ESL consortium. What does mean? This is a scientific network officially endorsed by the ESL. In this regard, we have different commitment and collaborations with the ESL. We are also part of the Global Cholangiocarcinoma Alliance that has the primary objective to establish a global voice in cholangiocarcinoma through community collaboration with the vision to raise the awareness of cholangiocarcinoma in order to improve prevention, survival, and the quality of life of patients globally. So in summary, there are multiple possibilities of collaboration within ENSCA and globally, which include the identification of unmet needs in cholangiocarcinoma, the comparison of cholangiocarcinomas in the different continents, to improve education and awareness, the facilitate of global sharing of experience and knowledge, the creation of working meetings and master schools, to improve the prevention in non-risk factors, improve also social media campaigns, the promotion of the creation of patient association in different countries, the improvement of search of funds for research, the increase the lobby to help change policy, the stimulation of interdisciplinary collaboration, as well as the recognition of reference centers of excellence in the study of cholangiocarcinoma. Also, we have multiple possibilities to unify the nomenclature, the diagnosis and the treatment, the acceleration to the access of therapies, and also to internationally validate biomarkers or therapies, among others. Finally, I would like to thank the different members of ENSCA, as well as all the different supporting institutions, including the European Union, the ESL, the Spanish Association of Gastroenterology, the Global Cholangiocarcinoma Alliance, the AMMF, and the Cholangiocarcinoma Foundation. Thank you very much for your attention.
Hello, I think I'm on. So I've been asked to speak from a North American perspective about mechanisms to promote translational science in cholangiocarcinoma and about opportunities for collaboration in particular. My talk is geared towards scientists in the field and those just entering, as well as to the concerned public who wish to know about how the field is moving forward together and where there are opportunities for further synergy. When we think of collaboration in biomedical research, we have in mind a number of elements. We think of working in cross-institutional consortia where large amounts of data can be pooled. We think of sharing biological samples or model systems. And we think about the formation of multidisciplinary teams where diverse expertise is needed to be harnessed to solve complex problems. Although we have much more to do, the cholangiocarcinoma community displays each of these elements. Moreover, there are exciting technological platforms that are beginning to serve as amplifiers for collaborative research. In areas such as cancer research and more immediately translational cancer research, where findings can potentially affect patients' lives, there's a very significant impetus for teamwork. Moreover, large programmatic efforts are underway, several of which you've already heard at this conference. And so fortunately in the academic world, there is an evolving view that collaborative research needs to be fostered in all research fields, which further lowers barriers. And I'll just say in brief, there's a recognition for the need for multidisciplinary research teams, and that's really being recognized about how the NIH and other funding bodies support research. So there's a favorable view of co-investigator grants, like co-primary investigator R01 grants, and large programmatic grants that invite teamwork, and I'll describe that in a moment. As well as just more practically, there's sort of a revised recognition within academia that collaborative scientists should be promoted. Then there are a recognition that some very large-scale technology programs require quite extensive investment, but can lead to really transformational data sets for the community. The TCGA, the C-Bioportal as a platform to interpret that data, and various repositories that I'll be speaking about. In addition, at this meeting, you've heard a lot about, from Rachna and others, the real power of cooperative oncology groups making what would otherwise be impossible trials possible. Just to further enhance the importance or emphasize the importance of collaborative research for cholangiocarcinoma, we have a relatively rare cancer type overall, but with extreme biological and genetic diversity according to risk factors and geographic regions. And so really to have a comprehensive view of the subtypes of this cancer, certainly we need to pool resources and ideas. I just wanted to highlight the evolving programmatic support for cholangiocarcinoma research. So in America, there's a specialized program of research excellence program, or SPORE program, that really tries to catalyze translational research teams within institutions, but then to integrate them into a national research community where live or real-time research updates are shared. And I'm happy to report that now at least 2 that I know of SPORE programs have as one of their major projects a cholangiocarcinoma project. So a project that Lipika Goyal and I and others at Harvard and Dana-Farber are involved in relates to FGFR-driven cholangiocarcinoma, as well as a recently funded liver SPORE program in the Mayo Clinic. And in common to those both projects is a desire specifically to nominate and optimal therapies for FGFR fusion-driven cholangiocarcinoma. So now I'm going to highlight some clinical collaborative work that's really exciting and only brought together by this terrific community of physician scientists that we have here. And so many of you will know about this cholangiocarcinoma in the young working group, which has this very broad cross-institutional team, which members are listed here. And this team was able to quickly gather a set of patients, again, representing both the typical presentation of cholangiocarcinoma in those above 50, as well as a reasonable sized cohort of younger patients. And this work really led to an appreciation that the biology of cholangiocarcinoma is quite distinct in different age groups. And again, motivating again attempts to devise therapies that are much more specific associated with these different subpopulations. And then finally, you've heard at this meeting some details about the International Cholangiocarcinoma Research Network, which again is now going really to most of the continents in the world, really to understand the heterogeneity across populations of cholangiocarcinoma. So now turning to some practical aspects of translational research, the first being collaboration around genomic data sharing, which can be a challenging area both for regulatory reasons and other bottlenecks. But many of my colleagues, Luca Goyal and others, have really been instrumental in overcoming some of these bottlenecks. And I'll show one study in that regard. And this is an attempt to harness a great deal of circulating tumor DNA to be able to understand the genetic diversity of this disease across populations, and to be able to really predict how effective ctDNA analysis will be to track disease across populations. And again, this was only possible by a large consortium generously agreeing to work together and pool samples, and they have a very exciting paper underway. Other work that you've seen from Lewis Roberts has assembled, again, a fantastic team to do genome-wide association studies, again, trying to decipher hidden genetic underpinnings of cholangiocarcinoma that, again, give us a better understanding of etiology and hopefully to predict vulnerable populations. And again, this is just an image of many of the collaborating groups here that Lewis has assembled. Another extremely successful collaborative endeavor across all cancer types has been the Cancer Genome Atlas. And of course, this is well known within the community. This began studying relatively common cancers using a very set of molecular analytic programs, using experts who were plugging in their expertise across cancer types. So again, there was no need to sort of reinvent analytical platforms or technologies, taking advantage of this NCI-supported Cancer Genome Atlas. And this has been geared on a smaller level towards cholangiocarcinoma. This was in part of their Rarer Cancers initiative, and it certainly gave some insights into the heterogeneity between cholangiocarcinoma patients and predictions about how to better understand the etiology of subtypes of cholangiocarcinoma. A development that we're very happy with within the cholangiocarcinoma network has been to try to work more closely with a data visualization portal, or CBio portal, developed at Sloan Kettering. And so this, again, well known by the cancer research community as a portal that makes all of this genomic DNA data that's generated by the TCGA and other programs readily accessible in a way that allows much more analysis without the need for sophisticated computational training. And we're very pleased that, again, Nikki Schultz from CBio portal presented yesterday, but we're very pleased that through the Cholangiocarcinoma Foundation, Nikki is able to turn his efforts for CBio portal to integrate many more available data sets into the CBio portal, data sets focused on cholangiocarcinoma, as well as to build in new features that I'll talk about very briefly in a moment, that are specifically geared towards improving the translational and basic science of cholangiocarcinoma research. So an effort that I'm involved in involves a multi-team effort for model development, both making genetically engineered mouse models representing all of the most common genotypes and eventually even the rarer genotypes in genetically engineered mouse models, as well as receiving the generous donation of tissue from patients to develop patient-derived samples. And this is to address bottlenecks in model availability. And as part of this consortium effort, working with some of these big analytical pipelines, such as the Broad DepMap, which uses functional screens to understand essential genes that are potential new targets for therapeutics, as well as the cBioportal to integrate these larger data sets into a way that's much more accessible. And then the third element is to make these models publicly available through the NCI patient-derived models repository, and for the genetically engineered mice, Jackson Labs, which will be distributing the models. So this is just a very brief summary of a development that is now being publicly presented. This is a cell line collection representing most of the important driver genetic mutations in cholangiocarcinoma patients. It's a cell line collection that has been supported in large part by Target Cancer Foundation with investigators across Boston and also outside the city. And the cell lines have been entered into the Broad DepMap. So again, giving this analysis of the essential genes that are in subsets of cholangiocarcinoma. And the cell lines are in the process of being deposited in the NCI patient-derived models repository, which is a very cheap way for investigators around the world to have access to this platform, but also to be able to access it in the context of having a very reproducible set of cell lines, so that one investigator using the same cell line in one country and the next is ensured of the quality control by virtue of the SOPs at the patient-derived models repository. And we're working with Nikki Schultz to develop new accessibility features in cBioportal to better mine the cholangiocarcinoma-specific data sets, including proteomics data sets and dependencies data. Again, and we hope that this is going to be a very widely used resource in the community.
Good evening. Welcome to everybody to the last scientific session that is about how to promote translational science, opportunities for collaboration. This is Luca Fabris. I am moderating this session. I am from University of Padua. And I would like to thank the organizer for doing this excellent job, because against the odds, it has been produced a very top quality Congress with many registrations from more than 40 countries. So congratulations for the great success of this event. I would also like to thank Jesus and Nabeel for their presentation. I would like to make a couple of questions. How this potential collaboration can be performed with the two countries? We have heard from the European experience and from the American experience that cover different aspects of what is a very big problem, that is the improvement of our understanding of this deadly. So, which efforts can be done to make this collaboration even stronger in a transatlantic way? Jus and Nabeel, what is your opinion? Which could be the efforts to improve this collaboration, starting from two different experiences?
So I think on a very simple level, perhaps a regularly scheduled kind of teleconference meetings where efforts can be coordinated. That was a beautiful presentation by Jesus And I can see a lot of ways that by communicating that and on regular communication, there could be opportunities for synergy just by setting up a regular meeting between groups.
Yeah, I also agree with you also, Nabeel, and thank you, Luca. I think, well, these kind of meetings help a lot to put the face to the others, because sometimes we, the Europeans, attend more to the European meetings and the American more to the American, although we also attend to each other, some of us. But I think at least the European network started as a group of friends trying to boost collaboration and to give visibility to this disease and to the research beyond this disease. But our idea and scope was to have a global international collaboration. We decided to generate it European first because we also have European grants specific for European countries, specific for collaboration between European countries. And this is an issue, the funding for collaborative proposals and projects. I think all of us have national grants and we collaborate each other, and this kind of network has helped a lot the promotion of collaboration between different groups. But since this is a rare cancer, we need to work more internationally in order to collect more samples, to be more ambitious in order to ask questions globally. And definitely we need to know each other, to have international meetings, conferences, but also we need to try to engage the foundations such as Cholangiocarcinoma Foundation, the AMMF, the Global Cholangiocarcinoma Alliance, and also the companies, because we need funding support to try to answer the big questions beyond. With the registries, we can have important knowledge. With the available sampling, we can get important resources for collaboration, for studies, but we need to find also ways to internationally find economic support to carry out this collaboration and accomplish the challenges.
I think that there are some research area, think about the Genome-Wide study, where the collection of samples from different countries is even more important. So I think that this is the way that we need to follow. And in your presentation, Jesus, I have appreciated the attempt to involve the Latin America countries to make cholangiocarcinoma registry in that area. But I think that we need also to reach out to other countries. Think about Africa, but even more important, the Asia, because Nabeel showed us very well that there are countries such as China, Japan, South Korea, Thailand, where cholangiocarcinoma is not a rare tumor. And we know that in those countries, the theology is completely different. Jesus mentioned about primary sclerosing cholangitis as a risk factor, and it is important to carry on the collaboration with the PSC study group. But I think that we should do some effort to reach out even to Asian countries, to focus even on the fluke infection etiology, for instance. What is your opinion on that? We need to work harder on this with this perspective.
Well, for our case, we are in strong and direct contact with Stacy Lindsay from the Cholangiocarcinoma Foundation and Helen Mormer from the AMMF and also with the Global Cholangiocarcinoma Alliance in order to create brainstorming, in order to promote international collaboration. And one of those is to try to engage different countries in Asia, for example, in the different registries, in the sampling, in the different collaborative initiatives that we have in Europe or in the United States. No? This is something that we need to take into a strong consideration, and then the Asia Pacific conferences can help to be in touch and actually meet those researchers and clinicians interested in cholangiocarcinoma in Asia, for example, and try to combine energies and promote collaboration. In Latin America, the funding of the Escalon project, which is a project between Europe and Latin America, has helped in the creation of the first collaboration with different Latin American countries. So we have on board several centers from Latin America within this group, but we are also going to present different collaborative initiatives on cholangiocarcinoma in the Latin American Meeting of Liver Diseases in the ALE, which was going to be in September in Brazil, but was postponed for next year. So the idea is to present to the physician and the researchers the different initiatives that we have in Europe and also in Latin America, and try to involve them in these new activities. By the way, Leo Fonseca, which is an oncologist from Sao Paulo, is very energetic, and he took with high enthusiasm this challenge, and he is helping a lot in the identification of key people and in the generation of this Latin American group.
I will say that there have been some quite successful efforts between America and particularly Thailand for cross, international research of the sort. So, for example, there's the TIGER consortium with the NCI and Thailand to understand liver fluke-associated cholangiocarcinoma, as well. The Target Cancer Foundation, Cholangiocarcinoma Foundation, has set up a models award to try to develop a set of cell lines and organoids from Thailand. And again, working in conjunction with North American researchers, it gives a lot of synergy there. So I'm pleased that those efforts are ongoing already.
A very good example also of this pan-international collaboration is the GWAS initiative that Louis Roberts is coordinating very well, and is supported by the Cholangiocarcinoma Foundation. Not only in the discovery phase, there are multiple centers from all over the world. Also in the validation phase, there are new centers in Latin America recruiting samples, or already with a good number of samples, and also I know that there are other countries that are in a good position to also provide samples. So these kind of projects supported by, in this case, by the Cholangiocarcinoma Foundation, but also by the AMMF, can help a lot to go beyond, beyond with important questions.
Yeah. To boost collaboration among groups, we need definitely the support by the big scientific societies dedicated to liver. And Jesus mentioned about the consortium with ESOL. It is very important. That can be paradigmatic of to widen the horizon of the study. Is there is anything similar, Nabeel, with the AASLD about this kind of support in North America? Is there any interest in creating a consortium with the American Association as well?
So I don't know.
Because I think that from this point of view, the European experience can be very important. What's about North America?
Yes, so I don't know about the ASLD specifically. What I've seen with them is certainly a seeming rising interest in cholangiocarcinoma as a disease. It was definitely not the top priority topic in meetings in the past, but it certainly seems to be an increasing interest in that organization. But I don't know about programmatic interests beyond for funding opportunities and so on. Yes.
Because of my
in this regard, I think that there are not specific interest groups in cholangiocarcinoma at the ASLD, but I know that the ASLD has a strong interest in the field. Actually, Greg Gors and Mario Strasabosco are organizing an intrahepatic cholangiocarcinoma meeting that unfortunately will be virtual because of the general problem. So I think ASLD is also taking important consideration on cholangiocarcinoma, and as well as ASCO, of course, and the ESMO. No? But I think at least ENSCA started mainly by hepatologists, people who work in the biliary field for many years in different biliary diseases and also in cancer. But our main society was the ESL, the Hepatology Society. No? That's why we arise to the ESL, no? Not through the ESMO.
There is a comment by Lewis Roberts saying that ASLD has a hepatobiliary cancer special interest group, and likely there is interest in collaboration even from this side. So thank you, Lewis, for raising this important comment.
Yeah, my only concern about that is that happened, for example, in ISL. In ISL before, liver cancer was only hepatocellular carcinoma.
Exactly.
And actually, I remember in 2015, in the postgraduate course on liver cancer, it was 1 day and a half, and there were 0 talks on cholangiocarcinoma. And the meeting was liver cancer. So that's why we arise, the ENSCA arise, to give visibility to this cancer in Europe and to promote collaboration and dissemination. And I am feared that this hepatobiliary group at the ISL, obviously because of the prevalence of hepatocellular carcinoma and the previous past, research, which is much more significant than on cholangiocarcinoma, the balance will be significantly different between the research beyond some hepatocellular carcinoma and cholangio. That's why I think it would be better to have specific groups on these different cancer and also collaboration and synergies.
I definitely agree with your opinion, Jesus, because we need to fight the idea that cholangiocarcinoma is still a child of a lesser God compared with cellular carcinoma. So I think that this is a very, very important point to raise. There is a nice question from Elisa Murjan that arise. Will this collaboration improve the access to new treatments for patients worldwide? I think this is very important because we are now facing with an evolving knowledge about molecular mechanism underpinning tumorigenesis and invasiveness in cholangiocarcinoma. But how we can move from this alteration to a real actionable target? Because I think that we need to bridge the gap in this about translation, the translational science in cholangiocarcinoma. We have many data, but so far still few treatments that can be really effective in curing the disease. What is your opinion, Jesus and Nabeel?
Nabeel, do you want to go first?
We need to search for a real actionable target. Because there are many mutations, but so far the gap with an effective treatment is still quite big.
Yeah, it's a big problem. Actually, most of the clinical trials on target therapy must to include a large number of centers worldwide because the proportion of the target mutation right now are very low frequent. So, and that's a point. So that's why we need to even more highlight the international collaboration that we need. We are part of one of those, and we have checked a couple of patients, a number of them, and still didn't find any of them with the target mutation. So we need to move forward. This is a very good, new therapeutic approach. We are everybody very, very happy and very have a lot of good feeling of that. But we need to promote new not only target therapies. I think also it's important to identify common key players in cholangiocarcinogenesis. So we and many other groups are trying to investigate or on common molecular mechanism altered in the majority of cholangiocarcinomas independently of the mutation. And there are multiple publications showing key transcription factors, for example, playing a key role like SOX17, like others, that need to have some attention, okay? Because I think the target therapy is very important, has opened a new field of therapy, but we need to also keep in mind that the cholangiocarcinogenesis process probably have, very, very probably have common pathological pathways, that can be potentially important for therapy.
I'd like to expand.
And I also want to highlight. No, sorry. Just finally, I want to highlight the point of Helen Morgan. So I think the action of the creation of the International PSC Study Group by Helen and supported by the Cholangiocarcinoma Foundation and other groups is very important. We need to make more dynamic this group. I think this is very important group, and we need to try to find the needs for collaboration within this group and try to see how we can give support to these initiatives.
So what I was just going to add is that I'm one of my biases is that you can learn a lot from genetically engineered mouse models, autochthonous models where tumors arise in their native context. And a lot of models that are generated for cholangiocarcinoma in particular have very strong drivers. And as we've just discussed here, that represents less than half of cholangiocarcinoma patients. But attempts to make models of the genetic subtypes where there's tumor suppressor loss and no sort of classical drivers can lead to hypotheses about immunotherapy combinations that might work, or a system that is readily amenable to genetic screening to identify other kinds of cell autonomous drivers. And so we've made a big effort in the lab to make models of these mutations like BAP1, but lacking, again, an obvious driver, BAP1 and other combinations, to establish this kind of suite of tools that mimic the heterogeneity between patients, including the unfortunate subset of patients where there's no actionable hypothesis based on genetics. So I hope that's one way that will at least nominate potential targets for these people.
I also would like to add that it would be very important to try to find collaboration with the pharmaceutical companies once we identify a good target. That I'm not talking about a mutation, I'm talking about a key driver gene which play a role in cholangiocarcinogenesis. And by experimental models, we identify that it's a key player. So we need to find collaboration from the preclinical to the clinical. So we need interest from the companies to try to identify those nice studies at the preclinical level that identify key players and try to set up new drugs or translocate these drugs that are available into the clinic. So this gap is missing. It's missing in whole Europe, and I guess that also in the States. So this is very important if we want to move forward from the preclinical to the clinical.
I think that's a good point.
And probably brainstorming meetings with pharma would be important.
Yes, I will say that it actually is quite pleasing in the States that there is a real will to invest in cholangiocarcinoma research from pharma here, beyond just the very obvious targets. And I think there's an appreciation that it's an area that there's not nearly as wide expertise as there is in a common cancer. And so there's a very high appetite for certain pharmaceutical companies to have real academic partnerships and to do some exploratory work. And of course, the next step also would be more drug development. But I am pleased that there's an appetite despite the relative rarity. So I am optimistic about that. But I think your point is well taken, that the more of that, the better. Will accelerate things more.
Thank you, Nabeel. Other question from the chat? Just me to have a look. Yeah, the last point is about the opportunities that this platform can provide. And Nabeel mentioned that we need integrated multi-omics approach to know better about the categorization and the mechanism of the disease. And Jesus in the ENSCA platform mentioned about the virtual slide tool that I think is a very good opportunity to merge samples and to analyze, to compare them in unified view. What is your opinion about these the technicalities that can improve the merge of the data within the two experiences?
Well, Nabeel has shown very nice data from those one, Catherine, about the merging of different omic data about the patient. So I think we all of us agree that we need multidisciplinary teams in order to face this cancer. We need genetics, immunologists, pathologists, oncologists, radiologists. It's very important to try to, because we are now generating a lot of information from different omic platforms, but we need to understand, to integrate them, and to really see what these provide, no, in terms of biomarkers, in terms of targets for therapy, no, and that's why this kind of bioportal tools and others are very important. In Europe, we are trying to generate also a collaboration from radiology, histology, and clinical, and then the generation of new data from the omic field. This is a project in which different people are involved, like Guido Carpino and others. So I think everything is complementary, and we can collaborate a lot in this, no, between Europe and the United States.
Nabeel, any comment on that?
No, I mean, I think that captured it well.
Okay. There is, I think, one of the last questions from the chat is from Judith Harris and Stacy Lindsay about the repurposed drugs in cholangiocarcinoma. I think that in many cancers we are facing this, that this is a very hectic area of research. What is the state in cholangiocarcinoma about drug repurposing initiative?
So I can describe one project in brief. Is the Broad DepMap PRISM project. So that involves a very large-scale barcoding of cancer cell lines and pooled screens with a very large number of compounds, and it's effectively a large-scale repurposing screen. And that's ongoing right now. And I think Stacy mentioned another effort in her notes here, but that's the one I know about, is one that I'm part of with the Broad, with about 50 cell lines compared to hundreds and hundreds of other cancer cell lines, so that a signal of sensitivity has a very strong reference. In other words, if sensitivity is seen in a subset of cholangiocarcinomas, it's not a random toxicity because we have a comparator of a large number of other cell lines. That's called the Broad PRISM program.
In my case, I'm not involved in any study on reposition of drugs, but I would like to highlight the data, retrospective data on aspirin, on the aspirin use and the statins, which show protective effects on cholangiocarcinoma development. I think these data were validated by different institutions, and I think are very, very interesting. And I think we should somehow prospectively start to do some studies in order to see if indeed they are valuable tools, not only to prevent the development under certain age or condition, also try to see what is the impact of to give adjuvant administration of this patient with aspirin or statin along any other oncological treatment. For example, it would be nice to see after surgery as an adjuvant therapy the potential use of aspirin. I think that could be interesting because aspirin do not allow the aggregation of the erythrocyte and the blood cells. So I think, and this mechanism is involved in the metastasis actually. So the molecular mechanism behind this is under investigation, but I think we need prospective studies, at least with these two drugs, no, aspirin and statin.
Yeah. I think these are common drugs that can be good examples learned from other cancer types of drug repurposing. So I think these are a way that we can pursue in the near future. So
yeah, also considering that NAFLD is a very important risk factor of liver cancer. I think the use of statin, at least in those patients, should be considered.
Okay. Thank you so much to Jesus, to Nabeel for this, for the very for the great presentation and the excellent discussion that we go through different points, and so very happy about that. Thank you for the audience for the question in the chat, and hope you have enjoyed this session, and congrats once again for the excellent result from this conference. Goodbye to everybody. Bye-bye.
Thank you, Luca. Goodbye. Bye.